Yes.
Okay, got it. Hello, everyone. My name is Caitlin. I'm from Sacramento, California, and I'm currently attending Pleasant Grove High School, and I'll be a senior this coming fall. My group and I will be presenting on EVs and pregnancy, and I'll be the first presenter for today. Our first slide is a table of contents to give you a quick overview of what we'll be presenting about in the articles and how long they'll be based on the slide numbers. And our first article will be talking about potential and challenges of isolating EVs, and we provided a quick outline of what we'll be talking about for this particular article. So to start off with the abstract, extracellular vesicles, also known as EVs, are particles that are naturally released from most types of cells, and these have gained an increase in interest among researchers because of their ability to provide a diagnostic information. EVs generally have very diverse characteristics, for example, their size, their content, features, and origin. And this type of diversity usually creates a layer of complexity when trying to isolate or characterize them. However, in this article, the researcher performed comparison among frequently used isolation techniques. One of the main reasons for an increase in interest regarding EVs is because of their cellular origin. In the introduction of this article, the content is explaining about the characteristics of EVs, which we'll do further debriefing on later in this presentation, but steering from that, the main reasons for an increase in interest of EVs is because of their cellular origin and biogenesis, which means the production of new living organisms. With an increase of interest, diving deeper into EV biology, researchers often find themselves with facing a lot of challenges. In this case, this challenge delayed their discoveries of how EVs can be used as a diagnostic tool. Throughout the article, the researchers aim to compare common EV preparations to find a good method for characterizing prostate-derived EVs. In this image on this slide, you can see a few methods, for example, density gradient centrifugation and ultrafiltration, and the results of each of them. So E through H are the different methods in the images that they produced, while A through D are the different results. As you can see, there's a very large gap of difference. So the researchers are trying to find the best method to characterize prostate-derived EVs. And for our next presenter for this particular slide is Alex Wu.
Hello everyone, my name is Alex Wu. I'm a coming junior in Santa Monica High School, and I'll start off with EV protein characterization. So, the absence of cellular contamination and most intense signals for the investigated proteins with special focus on PSMA, which is prostate-specific membrane antigen, and PTEN, which is phosphatase and tensin homolog deleted on chromosome 10, and lead to the decision on using ultrafiltration or UF in subsequent experiments to avoid the potential loss of EV populations for characterization. On the right, you can see a graph about protein concentration at increasing concentrations, showing the total particles per milliliter, and protein concentration. Now, about protein characterization, about immunomagnetic isolation, prostate-specific membrane antigen, or PSMA, were a main target of immunomagnetic isolation. One of the challenges delaying the use of immunomagnetic isolation for PSMA targeting is that PSMA appeared to have a weaker signal when targeted by PSMA beads. And to the right is a diagram of the steps of immunomagnetic isolation. Now, the next presenter for this slide about antibody microarray will be Wenfang.
Hello everyone, I am Wenfang Qi, and I am an incoming 11th grader at Venice High School in Los Angeles, California. So I will be talking about antibody microarray. So a spotted antibody microarray was developed to detect EV surface targets that might be helpful to pinpoint tissue relevant targets for immunomagnetic isolation. And the current design is made up of antibody spots in sets of 3 for measurements of 6 targets for up to 16 samples on each side. So as you can see in the image here on each slide, there's 6 groups of 3. and the discussion. So the size distribution, yield, protein concentration, EV-associated protein, and prostate-specific proteins from the different EV isolation methods are all compared in this study. And it was found that decreasing the concentration of particles was associated with decreasing protein concentration. And some challenges that were faced during this study was that it's unclear how a high G-force during ultracentrifugation might affect EV stability and function. And it's also unknown how many EVs are lost during the isolation process. Additionally, it's not possible to accurately assess the EV yield after isolation due to there being no method that measures the entire EV population. And high purity isolations of EVs seem to be accompanied with the loss of EV subpopulations, which means that there might be crucial information lost. And the next slide here will be presented by Alex.
Hello, everyone. My name is Alex, and I'm going to be an incoming junior at University High School in Irvine. So I'm going to be talking about the conclusion of our article. The combination of application and downstream analysis of EVs is an essential factor of guiding the choice of the EV isolation. UF, which is ultrafiltration, is a convenient method for initial characterization using and as a pre-step process in combination with the specific isolation using immunomagnetic beads, and to characterize EVs on an antibody microarray. The reason we do this is not only because it provides a fast, simple, and resource-friendly tool, it also thoroughly characterizes EV subpopulations. Heterogeneity of EV populations could be investigated with a specific isolation, and it might be a favorable technique to obtain disease-specific information from liquid biopsy. This slide provides a list of materials and methods we use in this experiment. Includes several important antibodies, nanoparticle tracking analysis, which is NTA, EV antibody microarray, and more. The next slide will be presented by Sophie.
Hi, I'm Sophie Zhu, and I'm going to be an incoming junior at Palisades Charter High School. And I will be presenting extracellular vesicles as critical mediators of maternal-fetal communication during pregnancy and their potential role in maternal metabolism. And this is a quick outline of what this article contains. Abstract. Pregnancy is one of the greatest metabolic changes undergone by the mother and the fetus. It's controlled by factors including hormones and molecules within EVs from the placenta, changes in the circulating levels and bioactivity, such as the capacity to induce changes in target cells of EVs originating from both placenta, have been associated with multiple pregnancy complications, such as gestational diabetes mellitus, preeclampsia, preterm birth, and fetal growth restriction. These complications are associated with maternal metabolic dysfunction and can cause short and long-term issues for both mother and child. The anabolic early stages of pregnancy generally include the storage of nutrients, while the catabolic later stages of pregnancy allow these nutrients to transfer to the fetus. This coordinates with a maternal insulin sensitivity decrease to nearly 56%, and an increase of 3 times the pancreatic insulin secretion. However, soon after removal of the placenta, reversal of insulin resistance has been reported, which implies that placental hormones play a role in modulating insulin resistance during pregnancy. While separate studies have shown that other factors, including TNF-alpha, may also affect insulin sensitivity, growing evidence shows that the concentration and bioactivity of placenta-derived EVs affects maternal metabolic regulation. A broad classification of EVs and EVs heterogeneity. EVs are membrane-enclosed vesicles originating from cells, including the human placenta, and carry specific cargo to their target cells to elicit reactions from recipient cells. Different EV origin and release mechanisms are the cause of EV subtypes. However, Different subtypes are difficult to distinguish from each other as they share physical and biochemical characteristics. This causes considerable heterogeneity in the field. Broad classifications of EVs include exosomes, which are about 50 to 100 nanometers, microvesicles or microparticles, which are about 20 to 1000 nanometers, and apoptotic bodies, vesicles, or blebs, which are about 1 to 5 micrometers. EVs that can be characterized by origin or disease have also been given names, such as large oncosomes. Small EVs have diameters that are less than 100 to 200 nanometers, and for the purpose of this section, we'll refer to exosomes. Medium-large EVs have diameters that are greater than 200 nanometers, and we'll refer to microvesicles. EVs during pregnancy. Pregnancy is characterized by higher concentrations of circulating EVs, along with other placental hormones and mediators. Pregnancy-related complications such as GDM and preeclampsia have been associated with small EV concentration and bioactivity changes. Many trophoblast-deported particles have been observed in maternal circulation, and these include placenta-derived EVs described as syncytiotrophoblast microvilli, and higher levels of STBMs have been associated with early onset preeclampsia and increased vascular dysfunction. An example of this is that syncytiotrophoblast-derived small EVs have been described to carry placental microRNA into the maternal circulation. Placental EVs have also been separated from other EVs using antibodies, the most common being against the placental alkaline phosphatase (PLAP) enzyme. These included using the NDOG2 antibody against the PLAP antigen. Researchers have also used fluorescent nanoparticle tracking analysis on Q-dot-coded anti-PLAP antibodies and discovered that placental small EVs are about 10 to 25% the total amount of circulating small EVs during pregnancy. Despite there being substantial evidence of changes in EV levels during pregnancy, the possible functions of circulating EVs during both normal and complication of pregnancies is still unclear. EVs in metabolism. Metabolic homeostasis is maintained by interactions between tissues. Changes in the extracellular environment, such as hypoxia or oxidative stress, triggers an increase in the release of EVs, which then leads to changes in the contents of cells, such as endothelial and kidney cells. It has been established that cardiovascular complications are associated with metabolic diseases. Cardiac stem cells tend to release more EVs in times of oxidative stress, which helps inhibit apoptosis. Thus, cardiac stem cell-derived EVs have been found to protect against oxidative stress and ischemic heart disease. Hepatocytes are liver cells, and their EVs have been shown to contribute to inflammation. Studies have shown that small EVs from hepatocytes and skeletal muscle cells regulate the beta cell adaptation in states of insulin resistance. Obesity also plays a major role in metabolic diseases and inflammation of adipose tissue. Maternal obesity is associated with pregnancy complications such as preeclampsia and preterm birth. Studies have found that small EVs from adipocytes communicate with macrophages, which can cause inflammation and insulin resistance. However, studies have also shown small EVs from adipose-derived stem cells have a protective role and promote browning in white adipose tissue. This shows the intriguing roles of EVs in mediating between cells and tissues and regulating glucose homeostasis. Metabolic disorders are connected to changes in EV amounts and their content, along with effects on target cells. For example, diabetic patients tend to have higher levels of circulating EVs, and gastric bypass surgery alters microRNA that targets insulin signaling. And these next two slides present some examples of findings from studies on EVs and metabolism and the conditions related. These few are all related to GDM, and they have effects on the release of cytokines, glucose uptake, and cell migration in skeletal muscle. And for these, the adipose-derived EVs both have been found to cause insulin resistance, and tests done with EVs from mouse quadricep muscles have been found to change the expression of genes in the cell cycle and induce insulin resistance. EVs in maternal metabolism in gestational diabetes mellitus. GDM is characterized by insulin resistance in skeletal muscle tissue and inadequate insulin secretion. High levels of total circulating and placental small EVs are present in GDM, and they have increased pro-inflammatory effect on endothelial cells. The differential expression of proteins and microRNAs in EVs reported in women with GDM were identified to affect insulin sensitivity and target insulin and glucose regulatory pathways. Studies have shown that higher levels of dipeptidyl peptidase 4 found placental EVs in GDM women decreases insulin secretion, and this reaffirms that placenta-derived EVs play an important role in the regulation of glucose homeostasis. Tests were also done on mice to show the role of GDM small EVs compared to small EVs from healthy pregnancies. In these tests, mice infused with GDM small EVs showed changes in the microRNA profile, insulin secretion, and insulin signaling. Since obesity plays a role in insulin resistance, women who are obese or overweight have a higher chance of developing GDM, and there is a positive correlation between circulating levels of small EVs and maternal BMI. Together, EVs play important roles in regulating maternal metabolism, and more insights can lead to the development of biomarkers for monitoring pregnancies. Conclusion and future directions. The placenta and EVs derived from it play important roles in communication between the mother and the growing fetus and may be indicators of physiological changes during pregnancy. The concentration and content of EVs change across gestation and in complications of pregnancies, including GDM. The potential of EVs in changing extracellular environments, inducing biological responses, and in cell-to-cell communication is receiving great interest in the field. Much of the current research is focused on EVs and metabolic disorders, especially as the mechanisms through which EVs regulate metabolic pathways is still unknown. Overcoming these gaps in knowledge may improve our understanding of reprogramming mechanisms in pregnancy and their related disorders. Thank you. And Alex Wu will be presenting bioactive DNA from EVs and particles next.
Okay, I'll be talking about extracellular vesicles and particles and their bioactive DNA. So I'll be going over a brief introduction, extracellular vesicles and particles, EV/EP DNA, and a conclusion. Extracellular vesicles and extracellular particles have been found to carry molecular biomarkers and mediators of intercellular communication. Most investigations on said extracellular entities focused on protein, lipid, and RNA, and because of this, little is known about them. In this review, we will provide an overview on the roles of EV/EP DNA as a functional mediator and molecular biomarker in various pathologic states. Cells are constantly releasing a large amount of molecular material used for intercellular communication. We can non-invasively access these extracellular components from biofluids to serve as molecular biomarkers. Extracellular DNA can release from apoptotic and necrotic cell death, as well as secretion in the form of nucleosomes and other nucleic acid protein complexes. Okay, large vesicles, extracellular vesicles, and particles. Large extracellular vesicles have a subpopulation called apoptotic bodies, and they are the largest cell-secreting vesicles at sizes larger than 1,000 nanometers. Microvesicles range from less than 100 nanometers to more than 1,000 nanometers and are classified as large EVs. Small extracellular vesicles have escort pathway regulatory factors that may influence functional differences between small EVs and other vesicles and particles. Until high-resolution separation of small EVs can be optimized, our understanding of small EVs will remain unclear. This diagram showing a size comparison of EV subpopulations to viruses, bacteria, and platelets. This is a diagram showing large EVs, small EVs, and extracellular particles, as well as their molecular markers, origins, diameters, and subpopulations. This chart shows EV/EP DNA distribution with each disease model, their source, EV/EP, and DNA origin. EV biomarkers have the potential in monitoring disease progression and predicting outcomes to treatment in cancer patients. A study in neurological malignancies by Garcia Romero showed that tumor-derived DNA could only cross the blood-brain barrier when associated with EVs. This study shows that EV-DNA could even be superior to plasma cfDNA for certain liquid biopsy applications in cancer. With its high potential as a target for liquid biopsy in a wide array of disease states, EV/EP-DNA will be a focus in the future of therapy and liquid biopsy. Therefore, the standardization of EV/EP-DNA isolation and analysis techniques is pivotal to ensure our complete understanding and utilization of this clinically useful entity. With slow, low-resolution EV isolation techniques rapidly becoming outdated, newer findings can be validated with newer, faster, and higher resolution technologies. Thank you. The next presenter will be Wanfang.
Hello everyone, my name is Wen Fong and I will be presenting the article on the effects of EVs on placentation and pregnancy disorders. So the sections I will be going over are the introduction, the identification of placenta-derived EVs, physiological functions of EVs in the placenta, diagnosis of pregnancy disease using EVs, the effects of EVs on livestock pregnancy, and finally the conclusion. So for the introduction, extracellular vesicles, otherwise known as EVs, are found in most body fluids and contain many types of RNAs, lipids, and proteins. EVs can be used to predict the physiological and pathological conditions of the cell of origin, and EV-derived microRNAs can stably exist in circulatory system, making them potential biomarkers for diagnosing pregnancy disorders. MicroRNAs help to regulate inflammatory responses in the invasion of trophoblasts in the placenta. And if the placenta does not adapt to the changed environment, pregnancy disorders may occur. Identification of placenta-derived EVs. So flow cytometry and nanoparticle tracking analysis allows for EVs to be detected in the maternal circulation, and they can also be used to differentiate between EVs from healthy and unhealthy pregnancies. So on the side here, you can see two images of flow cytometry and nanoparticle tracking. And some challenges of identification of placenta-derived EVs is that components of extracted EVs can vary depending on the method of extraction, and RNA can also be carried by non-EVs, which makes research even more difficult. Physiological functions of EVs in the placenta. So EVs can either have an immunostimulatory or immunosuppressive responses depending on the origin. So an example is the placental Fas ligand is released via EVs and suppresses T cells. And during pregnancy, the activation of T cells and the expression of inflammatory cytokines is correlated with pregnancy disorders, such as intrauterine growth retardation. And excessive immunostimulatory effects in the placenta are associated with pregnancy disorders. And placental EVs regulate immune responses and the invasiveness of trophoblast cells. So on the image here, you can see that there's an exosome that is affecting this endothelial cell. And this is just a chart of the examples of EV functions. So at the top here, you can see the Fas ligand and CD274, and the recipient cell is a T cell, and it basically just suppresses the T cell. Diagnosis of pregnancy disease using EVs. So I will be going over three diseases that can be diagnosed using EVs. So preeclampsia, preterm birth, and gestational diabetes mellitus. So for preeclampsia, individuals who have this disease had altered circulating microRNAs, such as microRNA 136, 494, 495, and 210, and they're all highly expressed in PE patients. And pregnant women with PE also have altered expression of certain proteins present in placental EVs, making them potential diagnostic markers. And for preterm birth, there have been 173 different microRNAs in EVs between preterm birth and term birth, recognized in recent studies. And they target the TGFB1 and TP53 signaling. And evidence from studies show that microRNA from the EVs of plasma can act as biomarkers to predict preterm birth. And for gestational diabetes mellitus, studies show that there is a larger increase in the concentration of placental-derived EVs in GDM pregnancies compared to normal pregnancies. The plasma EVs in women with GDM further increase the release of inflammatory cytokines, and microRNA patterns are also altered in GDM pregnancies. The effects of EVs on livestock pregnancy. EVs likely play a large role in livestock pregnancies. So for example, there's evidence that shows that the endometrium and placenta in ewes release EVs and microvesicles via the ureteric luminal fluid, or otherwise known as the ULF, which when isolated are shown to have contained different proteins and microRNA compared to ones extracted from cyclic ULF. And right here on the bottom is a chart. That shows some examples. So on the bottom here, you can see there's a cow, and for peripheral blood, it regulates inflammatory regulation, which is important for pregnancy. And the conclusion, during early pregnancy, the placenta has a mildly inflammatory and hypoxic environment. Various microRNAs and proteins from placental EVs play a role in pregnancy by regulating trophoblast invasions and the placenta's microenvironment. Studying and understanding placental EVs is an approach to diagnose and overcome these various pregnancy disorders. And next, we'll be presenting is Caitlin.
Hello, everyone. I'll be covering placental small EVs that focus on existing knowledge in the field and any current questions. Here I have a quick outline as to what I'll be covering for the subtopics of their article. With new discoveries and regarding how to control trafficking of EVs, it's helped create a new angle for understanding both local and distant communication among cells and tissues. In the following article, the content focuses on giving further explanations on existing knowledge and asking questions that hopefully can advance the research in EV biology. Starting off with placental trophoblasts, the article gives a brief description as to what they are, where they control gas exchange, supply nutrients, remove waste products, support the hormonal system, and act as immunological defenses. All these functions are necessary when it comes to the development of the fetus, as well as organized through communication of molecular cues involved in a healthy pregnancy. With these regulatory signals, further discoveries of large groups of EVs have provided a new angle for understanding signal communication. EVs generally transport nucleic acids, proteins, carbohydrates, lipids, and other metabolites that are all very different. Much like other cell types, placental trophoblasts create EVs of different sizes, but this type of process hasn't been thoroughly explained in previous research, but synthesis pathways like ESCRT pathways, also known as endosomal sorting complexes, required for transport, cell body fragmentation, which is a process where the cell splits into parts and then later those parts grow identically the same as the original cell. And other mechanisms, all these factors are said to take place in trophoblasts. In the article, it states that recent knowledge of EV cargo has been changed where placental small EVs are known to carry pro-apoptotic proteins, which are proteins that cause apoptosis, also known as controlled cell death. On the image on the right, you can see a specific EV subtype, exosomes, and the cargo that it carries. In addition, one main question in the field of EV biology mainly focuses on the possibility that certain cell-specific proteins like RNA and lipid molecules are selectively sorted to distinct EV subtypes. Selective sorting of proteins has been documented in EVs from other cell systems, while others are involved in protecting EV cargo against degradation. Degradation is a reduction of a chemical compound by splitting off one or more groups. With the small amount of cargo EV carries, it implies that the transportation of bioactive molecules to target cells would depend on trafficking and EV-specific delivery systems. With this said, EV trafficking can happen quite easily with neighboring cells because they're much close together, but when the cells are at a farther distance, it becomes more of a challenge. With this EV targeting distant cells, which can be known as different addresses in this case, is usually guided by unique barcodes, which are found on the surface of small molecules, for example, integrins. These barcodes are created by a combination of surface molecules, integrins, which are proteins that function mechanically and can sense whether adhesion has occurred. This overall process is still under wraps, but it is likely that molecules unique to the EV donor cells are required for specific recognition by target cells. The image on the left illustrates the process of creation of EVs with surface budding. With deeper research regarding the interaction of EVs with target cells and the process of barcode recognition, this will be necessary to understand more about EV trafficking and targeting. Moving on to EV entry. Some mechanisms like endocytosis, which is a process where cells absorb external material by engulfing it with the cell membrane, and fusion of EVs with the plasma membrane promote EV entry into target cells. Some of these processes can be intertwined with cell-specific recognition systems, trigger a surface molecule, or interact with the activation of target cell receptors. It's said that EV uptake is generally believed to impact the cell's physiology, which is a branch of biology that talks about how living organisms and their parts function. The article provides a few examples in the field of cancer, metabolism, and aging regarding physiology. In regards to diseases, many are known to come from abnormal EV production and packaging by the donor cells, trafficking, miscommunication, or abnormal uptake or processing by target cells. The image on the right illustrates the maternal placental circulation and the different EVs found in the placenta. Placental small EVs have been found to be involved in a number of gestational, also known as development disorders, and many are based on the connection between the diseases or abnormalities in small EV number or cargo molecules. In conclusion, having a final deeper understanding of EV biology has led to an acceptance of the presence of diverse EV subtypes supported in vitro and vivo testing. Vitro meaning within the glass, so test tube experiments using labware, while vivo testing is where researchers test a whole living organism or cell. However, with rapid progress, that doesn't mean that research must come to a stop. There are much more information that is unknown, which is why the article ends with a few aspects that can help further advance the field of EV biology. Thank you. So the next presenter focusing on trophoblast invasion is Alex Mung.
Hi everyone. In this section, I will be talking about trophoblast invasion, lessons from abnormally invasive placenta, AIP. Below is just a brief overview of what I'm going to talk about. First, trophoblast introduction. There are several reasons why trophoblast invasion to the uterus is important. For example, in order for fetal placenta to develop, trophoblast invasion to the uterus is required. Several essential functions of organs, such as establishing an adequate blood supply to intervillous space, controlling maternal fetal immune tolerance, and more. From the diagram at the top right corner, we see that in a normal pregnancy, we see blood flowing from the maternal placenta through the umbilical cord to the fetus site. Also, there are extravillous invading trophoblasts from the fetal site that will invade through the maternal arteries to the maternal site, potentially reaching the myometrium. This invasion is essential in providing adequate fetal oxygen and nutrients. In this slide, I'm going to talk about comparing preeclampsia to abnormally invasive placenta, AIP. Preeclampsia is the kind of disease that results in shallow trophoblast invasion and unconverted narrow spiral arteries. The situation is also called underinvasion, which contributed to insufficient amount of trophoblast invasion to the myometrium, causing a fetus to have inadequate supply of nutrients and oxygen. The second type of disease is placenta accreta, also known as abnormal invasive placenta, AIP. In AIP, there is an overinvasion of trophoblasts to the mother through the uterus. Types of AIP include increta, accreta, and percreta. From the graph, we can see an increase of placenta accreta system, PAS, from left to right. This meant that percreta, which is in the furthest right, has the most trophoblast invasion. The two risk factors of AIP are placenta previa and some form of uteral myometrial damage. Placenta previa is a disease where the placenta of the baby partially or totally covers the maternal cervix. The uteral myometrial damage is simply a damage, for example, a scar or tear to the uteral myometrium, which is the muscular outer layer of the uterus. Below are two experiments conducted posited by numerous scientists, a cesarean section have been argued by many that contribute to the presence of AIP, since scars provide a less resistant route for trophoblasts to permeate through. The first experiment is conducted by Garmi. In this experiment, uterine damage is used as a causative factor. The scientists measured the trophoblast invasion in the presence of a decidua, the thick layer of modified mucous membrane, which lines the uterus during pregnancy and is shed after birth. The second experiment is conducted by Hsian-Rong and Perest, and their experiment. They propose that cesarean scar dehiscence are the primary reasons that causes the horizon of placenta increta and procreta. This dehiscence causes a scar, which results in entry of chorionic villus tissue into the myometrium. There is evidence that changes in the invading trophoblast are associated with absent decidualization is obvious in ectopic pregnancy. In this type of pregnancy, two significant traits of extravillus trophoblast show that it has an intrinsically invasive nature and that it is able to differentiate in the absence of a decidua. Deriving from this conclusion, potential risk factors of pathogenesis of AIP include increased trophoblast invasiveness and altered axis. One counterexample, however, may be that of many pregnancies in which the presence of placenta previa in subjects who have multiple cesarean section does not result in AIP. In a diagram on the right, we can see that in a normal circumstance, very few extravillus trophoblasts reach the myometrium. However, in a situation where there is a scar, a significant increase of extravillus trophoblasts invaded the myometrium, and there is an even bigger invasion of trophoblasts in the presence of an altered decidua. I'm going to use an experiment to explain this section. In this experiment, researchers examined first trimester CTB, cytotrophoblast, and EBT, extravillus trophoblast, in order to contrast them to discover the underlying differences that indicate the existence of an EMT, an epithelial mesenchymal transition, as a mechanism of differentiation. The procedure is to examine the expression in CTB and EBT of 84 genes associated with EMT, to determine if changes in these genes could provide evidence of an EMT in the process. The result of this experiment demonstrates that many of the gene expression changes were characteristic of an EMT. Based on this result, researchers analyzed that these different results prove the different variations of EMT. Data also supports that CTB-EBT differentiation process is a unique type of EMT, separate from those identified such as gastrulation and metastasis. Also, since it occurs so early in human development compared to EMT types 1 to 3, speculations that CTB differentiation may be the molecular archetypal EMT, a type 0 EMT. This section is about EMT status in the third trimester. For similar to preeclampsia, AIP is generally only identified and accessible in the late second and third trimester. In the first trimester CTB to EBT differentiation, the process is accomplished via an EMT, with EBT displaying a metastable mesenchymal phenotype. In the third trimester, however, many of the gene expression changes observed in the first trimester were much reduced, as you can see from the two graphs. From the above two tables, one can clearly see that in the left, which is the first trimester, the fold changes value were significantly higher than the fold changes value in the right, which is the third trimester, regardless of whether the gene is downregulated or upregulated. Even though incidences of AIP pregnancies are steadily rising, the occurrence rates are still considerably low. After analyzing AIP from cases where histopathological analysis has confirmed the presence of either increta or procreta, results show that normal and previa EBT are very similar. The table on the right shows the result of the experiment. Comparing this table to the previous third trimester table, we can see the increase MMP2, TGFB2, and ZEB2 gene. After analyzing multiple EMT master regulator transcription factors, results showed that only ZEB2 gene, which is zinc finger E-box binding protein 2, displayed characteristics which match both the invasiveness of cells in vivo and with EMT status.
ZEB2 genes provide instructions for making a protein that plays a critical role in the formation of many organs and tissues before birth. Following three pieces of data are essential in the derivement of the conclusion. ZEB2 transcription factor was responsible for regulating progress of trophoblast cells through the EMT. Three pieces of evidence are: in first trimester trophoblast, the B2 gene expression is increased by almost 200-fold in EBT compared to CTB. In third trimester, it drops back to the level below that of third trimester CTB. So the conclusion. Recent research shows that changes in the decidual myometrial environment surrounding the trophoblast may have major effects on trophoblast function. The data throughout this section provides a clearer definition of EMT as a mechanism of CTB, EBT/EBT differentiation. The EMT also provides a solid mechanistic basis for the abnormally invasive pathologies and for investigations of the defects, trophoblastic or uterine, which are casual. Thank you for listening, and this is our acknowledgement. We would like to express our gratitude for Professor Zhen and Dr. Zhu for giving us the opportunity to learn and gain valuable experience from volunteering in this research group. We look forward to working with everyone very soon. These are some of the references and websites we use to assist us during our presentation. Thank you everyone for listening.
Okay, so floor is open for questions. I would say, why not our juniors go ahead, raise a question to this 1, 2, 3, 4, 5 speakers. I think in brief, I think they did an excellent job. Okay, so I will comment at the end. But well, any questions from audience?